Hi, this is Miss Litton, and this is 7th period, and these are wonderful honors bio students. Say hi.
Hi.
And we are doing a little double Quizlet, and we are doing it over chapter 5, which is about cell division. And the three big things there are cell cycle, mitosis, meiosis, okay? And then in chapter 22, we're just doing the very first section of that, and that's on development, and that we talked about last class. So the first thing we want to remember is we want to remember our cell cycle, and that's kind of the life cycle of a cell. And so we have the M stage. The M stage includes the stages, in this case, of mitosis. And when you look at each of those stages, you should be remembering what are the chromosomes doing in prophase. We'll review it, but in your mind, just to yourself, think about what's happening during prophase, metaphase. In a metaphase, something's always in the middle. Good. On an anaphase, something's always going away. And on a telophase, you're reforming the nuclei, right? And then cytokinesis really should be superimposed on top of telophase, because cytokinesis is about not the nuclear division, which mitosis technically is, but everything else. So that's called what again? Yeah, division of the cytoplasm and cytokinesis. Okay? Then what happens is after the mitotic stage, the M stage, the nucleus kind of just looks like a solid circle again. And you would have like a dark, maybe nucleolus reformed again inside of your nucleus. And it looks like it's not doing squat, but it's really doing a lot, okay? And the first stage of this next part right here, okay, all of this right here is called interphase. And interphase consists of three parts, G1, S, and G2. And there's certain things that happen during G1. First of all, you had one cell and you just went into two. So those two cells have to get bigger. And you have to make more of the cellular components, so you make more organelles. You don't need to make more nucleus because you already doubled up the nucleus earlier, right? Right? So they already have their nucleus. You just need to make all the other parts. Then the S stage of interphase, what does S stand for? Synthesis. What are you synthesizing? DNA. Okay? This is when you form your sister chromatids held together by a centromere. So you have 2 copies of every single chromosome in that cell. Do you see them as rod-shaped chromosomes at this time? No. It just looks like something you pulled out of your what? Hairbrush. Right? You can't see the chromosomes. They are doubled up, but you just can't tell visually with your eyes. Okay? Next we go into the G2 stage. What are you primarily making in the G2 stage? Proteins. Okay? Now, why would you double up the DNA in the first place? Because you're getting ready to do the M stage. And what's the M stage? Mitosis. You need 2 copies of the DNA because in mitosis, the dance, prophase, metaphase, anaphase, telophase, that's all about making sure your chromosomes are all wound up on chromosomes. They're lining up in the middle. You're separating them. Each cell gets their own nucleus, and then you divide the cytoplasm. That's basically the M stage. Now, one thing we can see in here is G0. G0 is like an off-ramp of the cell cycle. It's like, I'm done. And at following or in the middle of G1, you take the G0 off-ramp, which means I'm not going to synthesize my DNA. I'm not going to make more proteins because I'm not going to do what?
DNA synthesis?
Yeah, but why am I not doing DNA synthesis? Because I'm not going to do mitosis. I'm done doing mitosis. Exactly. Okay? And then I gave you this little diagram. Do you remember what everything is? Okay? Where should we, yes?
Does the cycle start with mitosis or does it start with G1?
A cell's life starts at the completion of mitosis. Okay, so you're done, so we're going to start, we could start anywhere on here when we're referencing it, but the beginning of that cell would be as soon as it finishes mitosis. So tell me again, what happens during G1? Cell grows, and what else? More organelles. So we're looking for more organelles. And did I do it? No. Because I'm a highlighter, because I'm so smart. There, now we made more organelles. What follows G1? S. So what are we going to see during S? That's right. Is it actually doing this? Would you see them as rod shapes? No. Just look like something out of a hairbrush. But those are sister chromatids. These two greens are exactly the same. Okay? Then what follows the S stage? G2. And these are abnormally large what? Proteins. Yeah. So we're going to make more proteins. Then you're going to go through the mitotic dance, which we'll review the steps of those in just a minute, and cytokinesis, and you're going to form two cells. And then each of these cells will undergo G1, S, G2, and then make some more cells. Now this is typical of somatic body cells. I'm twisting back into chapter 22, right? Watch me grow. Remember that question with the little boy? We watch him grow because all of his cells are doing this, mitosis. Clonal replication, exact same copy, no variety at all. If whatever cell you have, if you're a potato, you have 48 chromosomes in your cells, you're going to copy all 48 chromosomes, and all your daughter cells are going to get 48 chromosomes, and you're going to become a bigger and bigger potato. We okay with that? All right. Now, we talked about that the control of this, when it takes place, kind of equated it with if you're going to go fly an airplane. In order to actually get on the airplane and take off, first of all, you have to have a license.
Boarding pass.
Yeah, you have to have a boarding pass, right? A ticket. You're not all becoming pilots. Where are you? So you'd have to have a ticket, but that will get you in the airport having that ticket, but then you have to go through security. Have to go through security. And it's the same thing for the cell cycle, because we know there are plenty of instances where the cell cycle is out of control and it's going too fast and making too much. What do we call that when it's out of control like that? Cancer. Yeah. Or cells stop making more cells. So we want to regulate that so we maintain homeostasis. So the tickets are called cyclins. And you have to have M cyclins build up before you do mitosis. You have to have S cyclins build up before you do synthesis. That's like your ticket that says go on to the next stage. Because if you're wondering, and did I have you do a cell cycle POGL? Was that in your POGL? Yeah, it was. Yeah. So the cyclins build up. Think of that as like your ticket. Then you have these checkpoints. Here are 3 different checkpoints. The biggest checkpoint is right here in P51, but it's in the G1 stage, and it's checking to see, did you do mitosis correctly? Are you sure? So it's like a little stop sign, and make sure it's okay. Here, this checkpoint in G2 is saying, did you do the synthesis correctly? Did you accurately replicate the DNA? This one right here, it's in the middle of mitosis going, are you lining up the chromosomes appropriately? Because if not, we're either going to try to fix it or you're going to kill it. And killing cells is a normal part of development. Remember in development we talked about fingers, right? And what has to happen in between the digits? Apoptosis. So apoptosis, it's a balance between mitosis and apoptosis for normal growth, development, and repair. Okay? Questions, concerns on that? All right, so let's keep going. We talked about how to build a chromosome. When you look at this picture, you can see the double helix of the DNA up at the top. If I backed away farther from the DNA, it wouldn't appear as 2 strands. It would probably appear as what? 1 strand to me, because I'm farther away. And that's what's happening here. You're not as magnified. So here we can see this DNA. This DNA is winding on these protein spools called nucleosomes, and nucleosomes are built out of histone proteins. Then we're backing up a little bit more. These histones will twist together. It's like braids on top of braids on top of braids till we have a dreadlock. Okay? That's what's happening here. It's twisting on, twisting on, twisting on, and then we can see those rod-shaped chromosomes. Now, this right here is one sister chromatid, and this right here is the other sister chromatid. Never draw for me things like this in an X. It's not X. Okay, it's like this. It just looks like an X. Okay, I don't want you to miss any points about that. It's just two rods, and they are in this location. They're just like pinched together right there. Okay, but it's not an X. There's one chromatid, and here's the other chromatid. All right, questions or concerns? Ask your bio buddy. How you doing?
Pretty good.
Okay, now, we then had a discussion, come back to me. We then had a discussion about the term haploid and diploid. Okay, now each of you in your somatic body cells, you have a pair of chromosomes. One half of the pair came from your mom. She could give you chromosome 8. And your dad also gave you chromosome 8. That is a homologous pair. They are the same size, the same shape, and they code for the exact same characteristics. Now, one might say blonde hair, one might say brown hair. One might say blonde hair, one might say brown hair, but they're both coding for hair. Okay? That's why they're called a homologous pair of chromosomes. The sequence of genes on them is exactly the same. The variety, the alleles, those could be different. Okay? So this right here is not showing you a homologous pair. This is only showing you one chromosome. But because all in the S stage, every chromosome replicates itself, it appears doubled because it is doubled. This could be 2 copies of mom's chromosome 8. If I was going to draw a homologous pair, okay, I could say, okay, here's dad's chromosome number 8. This is the homologous pair. Follow? These are sister chromatids, and these are sister chromatids. When you do, big picture now, mitosis, do you care about the homologous pairs when you do mitosis? No. Every single chromosome lines up single file in the middle of metaphase because you want every daughter cell to have the full complement of all 46 chromosomes. Yes? So they line up single file, and the only thing you're separating when you do mitosis, the only thing you're separating are the sister chromatids. That's the only thing you're separating. When you do because mitosis is my arm, what? My leg, mitosis. That's in a body cell. Meiosis, okay? That's when you're getting ready for sex. That's only going to happen in special places called, what am I modeling now? Ovaries or testes. That's the only place you do meiosis. You still do an S stage. You still replicate all the DNA. You still make sister chromatids. But now the homologous pairs come into play. The moms and dads come into play, right? Because when you make sperm or eggs, you can only send one of this pair. Either you're going to send a chromatid from dad, or you're going to send a chromatid from mom. Otherwise, mutant Ninja Turtle, right? We got to get down to half the chromosomes. If every body cell in us has 46 chromosomes, then our sperm or eggs should only have how many in them? 23. So when little Mr. Happy's sperm, haploid, 23 chromosomes, hooks up with little Miss Happy, egg, haploid, right? Fertilization. What's the little cap on the sperm called? What's his name? It does. Acrosome, which digests the zona lusa, and you do fertilization. You have a diploid zygote. Homologous pairs, right? And then you have a diploid zygote that has the full 46. 23 in the sperm, 23 in the egg, now 46 total chromosomes. If it's a boy, the 23rd pair will be X, Y. If it's a girl, the 23rd pair will be X, X. We're going to learn about that in the genetics chapter. Okay? All right. So, when you talk haploid, diploid or diploid cells, the only haploid, diploid cells you and I have are sperm or egg. Plants can have more than that. We'll put a pin in that and talk about it another time. But for the purposes of humans and mammals, the only cells that are haploid are our sperm or our eggs. And a sperm that's haploid gets together with an egg that's haploid and forms a diploid zygote, which will grow up into a young baby. Okay. All right, if you remember that from the video. Yeah, remember that? Yeah. Okay, so as far as diploid numbers go, a tobacco plant has 48 chromosomes. That's its diploid number when it has all the chromosomes. So that means the sperm or egg of a tobacco plant, the pollen or the egg, would have only how many? 24. You got it. That's haploid, diploid. Okay? Now, you do mitosis for growth and repair. Okay, either getting bigger or repairing the tissues you have. Plants, we keep, like, our bone marrow can still do mitosis and things like that, but then, like, then you're done. Whereas plants always have meristematic tissue that can continue to grow. So when we looked at mitosis, we focused first on the stages. Remember, it's PMAT. PMAT, like a dog has a mat to pee on, PMAT. Prophase, metaphase, anaphase, telophase. And it's one solid continuous or fluid continuous dance. It's just that we're saying, hey, when the chromosomes are here, we call it prophase. When the chromosomes are here, we call it what? Metaphase. Got that? Okay, so prophase has a lot of stuff that happens during it. We can see here, remember the things we look like churros? What do we call them? Centrioles have doubled, and the centrioles, by the end of prophase, have moved to either end of the cell. And I told you, it's like if you're putting up a clothesline to dry your clothes, old school, if you didn't have a dryer. Those are, the centrioles are like the poles on either end, okay? Then the line across, that's what you see forming right there. Those are microtubules. Remember the cytoskeleton when we learned parts of the cell? Those are microtubules, okay? Then here, your DNA, which started off just looked like something out of your hairbrush, like we saw earlier when we made the dreadlocks, they start to wind up on each other, and you start to see these rod-shaped chromosomes. That all occurs during prophase. And also your nuclear envelope, we get rid of that. The nucleolus, we get rid of that so that the chromosomes can move about. Okay? What stage follows prophase? Metaphase. In metaphase, every single chromosome lines up single file. The only thing there are the sister chromatids held together by a center. They're single file, boom, 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 all the way down. Then in anaphase, you separate the sisters from themselves. Remember I said it's not an X, right? It's like this. And so the reason why this picture appears like these V's is because the chromosomes are getting grabbed onto and like pull apart, like they're getting pulled apart. Okay, so you're separating the sister chromatids. And that's anaphase. Telophase, you're now protecting those chromosomes that have separated in a nuclear envelope. The nucleolus starts to form. If it's an animal cell, you're forming a cleavage furrow there. Remember cleavage, a cleavage we think of like breasts, I get that. But a cleaver is like a knife that does what? Cuts, separates. So this is called a cleavage furrow. It indents and then like pinches off so you have two cells. Plants do this differently. Okay? When plants do that, they have to build a cell plate. All right? So let's do the mitosis song. What's the whole thing called? Mitosis. First stage, prophase. The chromosomes. In this case, how many chromosomes do I have? 2. 1, 2. And these would be what? Good. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. Asters, because it looks like an asteroid on the other end, right? Do you remember what part I'm talking about there? Asters. Asters. Okay? They meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Okay, remember, Evan, I just did this on Thursday, so this part is new. I redid the calendar. Remember how what I did is I added a third column of review resources. So if you look at that third column on the bottom, Mrs. Litton's 7th period support, I believe I have the songs there. Somebody checks. Do I?
Yeah.
Okay, Swedish.
Do you have like the videos too, or just like?
Yeah, the videos. Yeah, me doing them.
Okay.
Yes.
Are microtubules and asters the same thing?
Oh, very good. Microtubules make up the aster. Yeah, when you talk about asters, when you look right here, it's like all these radiating microtubules, that's referred to as the aster. Okay, now I feel compelled to tell you, there's a difference. When we do cytokinesis in animals, it's a cleavage furrow, whereas plants build a cell wall. There's another difference. Plants, remember, don't have any, you learned this already, they don't have any centrioles. They still have the aster, just no centrioles sitting in the middle. Why? I cannot tell you. Don't know. Plants don't have centrioles. Okay? All right. Now, then we reviewed the idea that when you want to make a baby, okay, you need to give them half your number of chromosomes. So instead of doing 1 division, we do 2. The second division looks exactly like mitosis, right? So the first division is the only thing that's different, and the reason why it's different is instead of lining up single file, you line up with your homologous pair, right? And that way, the first division in Anaphase 1, you're separating mom and dad. We called that getting a divorce, okay? You separate mom and dad. And remember, we said a couple of things can happen. Before Metaphase 1, right? What stage comes before Metaphase 1? Prophase 1. And what can happen during Prophase 1? I'm doing it with my fingers. Crossing over. They can exchange pieces of their chromatids, and that just increases variety. That's fun. Okay? In metaphase, each homologous pair can line up independent of all the other homologous pairs. It's still in the middle, but it's not like it's like all dad chromosomes on the left, all mom chromosomes on the right. It's not like that. They line up any which way, which also increases variety. Okay? So Anaphase 1, you separate all the homologous pairs, telophase 1, cytokinesis 1, prophase 2. Metaphase 2 looks just like mitosis. All the chromosomes line up single file. Anaphase 2, you separate the sister chromatids. And we'll go over that as well. And the reason is we are trying to form haploid sperm to hook up. You guys, whatever you're doing, you're distracting yourselves. A haploid sperm with a haploid egg because we're trying to form, this diploid baby, right? Diploid baby. And you have that life cycle you can look over in your notes, or the naked people. Remember, we went over that. And we talked about homologous pairs. If you hold up 2 fingers right here, okay, how many chromosomes do I have? 2. Okay? If this one came from my mom and me, then this one must come from my dad, right? This is homologous pair. What happens during the S stage? Okay? I still have how many chromosomes? 2. It's just that each chromosome consists of 2 sister chromatids, copies of themselves, right? And so in prophase 1, all 4 of these chromatids are going to hook up together in synapses, crossing over. Metaphase 1, they line up. Anaphase 1, separate the homologous pairs. In each of these 2 cells, I do anaphase 2. Anaphase 2 takes place in 2 cells. Anaphase 1 takes place in 1 cell. All right. Okay, and then we went through and we looked at the stages. We can see evidence here. It looks like they're crossing over right here. And sure enough, they did, because look, there's this guy's red and this guy's blue. Okay? Now, they lined up where it went blue, red, blue, but they could have just as easily have lined up blue, red, blue, red. Either way, that's independent assortment, and that occurs during metaphase 1. Anaphase 1, you're separating the homologous pairs from each other. And then you do telophase 1, okay, and you end up forming 2 cells. These cells are technically haploid, because how many chromosomes are in this cell? 2. How many did we start out with? 4. So we're down to 2. Yes, each of those chromosomes consists of 2 sister chromatids, but technically it's haploid because they don't have their homologous pair. Yeah, they don't have the other half of their pair. Each of those undergoes meiosis 2, okay? And then each of those, this could be a sperm, right? And in this one, is skip a stage? No, I did not. Okay? Where it looks like one picture is missing. Is a picture missing? No, they just didn't show that one. They didn't show Metaphase 2, Anaphase 2. Sorry, but that is in there. So when we ask, how can these puppies born from this mom and dad, how can these puppies be so different? What are our 3 reasons? Crossing over, independent assortment, and you never know what sperm is going to fertilize what egg. Okay, so let's do a meiosis 2 dance real quick. Okay? So, show me S. Okay, good. And what's the first stage? Prophase 1. Right now we're doing meiosis, right? The whole thing's called meiosis. We're going to do prophase 1. The chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, asters, synapses, crossing over. Okay? Then they meet in the middle at metaphase 1. They meet in the middle at metaphase 1. And to remind us of independent assortment, we're going to go, they meet in the middle at metaphase 1. Okay? Away at anaphase 1 to nuclei at telophase 1. Cytokinesis, interkinesis. It's the resting stage in between. Now we're going to do prophase 2, metaphase 2, anaphase 2. What are we separating in anaphase 2? Okay. Then telophase 2, cytokinesis, which I can't do because my fingers are attached. And then I will make 1, 2, 3, 4 cells. How many chromosomes are in each one of these cells? 1. How many did I start off with? 2. See that? So I'm down to half the number. All right, that is chapter 5. Okay. Interkinesis is the time between the two. Okay, now, so let's jump to, okay, we're like trillions of cells coming from one zygote, but yet we have all these specializations. And so we were trying to understand how does that happen ultimately. And so we talked about how haploid sperm will fertilize the egg. It has to digest the jelly coat using enzymes from the acrosome. It's going to burrow in and then ultimately fertilize that egg. And we talked about how right away, as soon as the sperm nucleus comes in contact with the cell membrane, when the sperm membrane comes in contact with the cell membrane of the egg, there's a fast block so no other sperm can get in, because otherwise mutant Ninja Turtle, right? And then there's a slow block afterwards. Okay? Then we broke down the whole discussion into the what and the how. The what is, we just learned the stages of development. We learned the stages of development and key characteristics of each stage. And one thing we walked away from in that process is knowing that the amount of yolk you have can affect how the cells and tissues move, because the yolk would get in the way. Let's start by just reviewing the stages. So here comes Mr. Happy Sperm, and he is what? Haploid. Here is the egg. She is also haploid. These were both made by what process? Meiosis, because we're getting ready for sex, which clearly they have just had. What is the cap on the sperm called? Acrosome, and it digests the zona pellucida, and you have fertilization, forming a diploid zygote. That zygote does the cell cycle so fast, it's like blows right through G1. It still does S and G2, but it does G1 so fast that those cells don't have time to grow. So we call that time cleavage, because it looks like it's just cleaving. So we go diploid, zygote. Then we go, what did I say to you? Cleavage, and just bounce it. Cleavage, and you form a solid ball of cells called a morula. Solid ball of cells. Bunch of cells, same size as our zygote. Then we pump in salt, right? Basically, those cells are secreting salt to the center of the morula. Who follows? Water, because water must flow from the hypo. Water flows in and blows it up to a blastula. And a blastula is a hollow ball of cells. What do you call the cavity inside, this hollowness inside? A blastocele. Good. Then we let out the gastrula, and now we have a layered ball of cells. At first, just 2 layers. What's the outermost layer? And the innermost layer? Eventually it forms a 3rd layer. What's the 3rd layer called? Mesoderm. We know what all the layers are going to become. All we have to do is memorize the outer and the inner, and then we default everything else to the middle layer. So what comes from the outermost layer of these cells? What comes from the outermost? You were like in it, like skin, nerves, islands. Good. And then what comes from the innermost layer? Digestive and respiratory. And the way we remember that is we make your gastrula, your layered ball of cells, the new cavity inside, because the blastocele, this cavity, got smushed away, right? Scientific word. And now we have a new cavity. The new cavity, it looks like an arch, so that's why we say archenterum. Say it. Archenterum. The opening to that new cavity is called a blastophore. Okay? Our blastophore became our anus. Okay? Other organisms, the blastophore became their mouth. Now, when we think about our mouth, there's only 2 systems that we can access through our mouth. What are those 2 systems? Respiratory and digestive. So that's why it makes sense to us that the endoderm is going to generate the digestive and the respiratory system and any associated organs and glands. We good with that? Okay. Now, we looked now at the last stage, so be your gastrula, right here. Pretend like your thumbnails. Remember this part. I hope that is helpful. I know it clicked off at one point, but hopefully you got all that. All right. Have a piece of toast.